Joa, hi. Unfortunately, this was the wrong intro video, but I think
that's okay. Ghent is a beautiful city.
You're at the right place.
I mean, frankly, I would prefer to be in Ghent than in Liverpool. It's lovely. I've been there many times when I lived in Belgium. So
right, awesome. Quick introduction: Joao is a scientist and futurist. After doing his PhD and after his training at the Harvard Medical School, he's now a professor at the University of Liverpool in England, not in Ghent. He's also a founder of Magellan Science Limited, which focuses on consulting on data analysis and longevity science. Very fitting to this time, his talk is titled "Winter is Coming: The Future of Cryopreservation." Joao, please take it from here.
Thank you. Well, thank you very much, Emil, and thank you for the invitation, and thank you for putting together this conference. I mean, obviously, again, I would prefer to be in Zurich as well, actually giving this talk and meeting you all personally, which is unfortunately not possible, but hopefully next year. And so, I mean, I'll start with a brief introduction. I know some of the people in the chat know who I am already, but I'll start with a brief introduction to those not familiar with my work and my ideas. And then I'll talk about a couple of different projects we've done, a little bit of work we've done in cryobiology, and also in more in the context also of cryonics. So I guess since when I was a very young child, this is me as a child, I realized that, you know, everybody dies, and we've got to do something about it. You know, the idea of death was just like this, you know, eternal oblivion. I mean, I'm not religious. I don't think there is an afterlife. I think that when you die is just like a eternal prison. So really the concept, I would say, scared me. It still scares me today, the idea of dying. So I wanted to do something about it. I wanted to, you know, let's try to fix it in some way. And so I started to focus on aging. So my day job, so to speak, and most of my work focuses on aging. So as Emil mentioned, I'm a professor at the University of Liverpool. I lead the Integrative Genomics of Aging group. And we do various levels of research on aging, combining both computational approaches and experimental work. So this involves, you know, systems biology, bioinformatics, machine learning, AI, but we also do dietary manipulations of aging in animal models, cellular models. We study cell senescence, and we do genetics of longevity in human cohorts and populations. So we do a large variety of work. I mean, if you're interested in our work, obviously, please visit our lab website. So my idea, I guess, when I started on it, I became a scientist not because I wanted to spend my days applying for grants and writing papers, but because I wanted to do something. I wanted to, you know, make a change, in particular at the level of longevity. I wanted to extend our lifespan. I wanted to cure aging. My dream from when I was a child is I want to cure aging. That's what I want to do in my career. And that's why I developed my academic research in this area. And well, as Emil mentioned, I'm also doing some work in biotech, which I won't go into detail today. I do think that once you start reading into it, although there's been a lot of advances in the field of aging, for example, in pharmacological manipulations of aging and genetics of aging, I don't think we're going to, or I think it's very unlikely, I wouldn't say it's impossible, but I'd say it's very unlikely we're going to cure aging in my lifetime. I think it's just a too complicated process, and our understanding of biology is too limited for us to cure aging in my lifetime. I mean, I wrote this paper some years back in rejuvenation research on the prospects for curing aging that goes into, you know, the more detailed discussion of this, and of course happy to discuss in more detail later on. The point was, okay, we're not going to cure aging. If you're not going to cure aging, then that means we're going to die. As between eternal oblivion and which is death, I mean, is there another choice? And that's why I became interested in cryopreservation and cryonics, because of course, cryonics, again, without going into a lot of detail, there will be other talks on cryonics. It gives a possibility of future life. I think there's limitations of cryonics and cryopreservation that I'll mention in a minute, but between eternal oblivion and death and a possible future life. Cryonics gives that possibility, even though, I think there's a lot of arguments, whether that's a high possibility or a very low possibility. I would argue it's actually a very low possibility at the moment. The reason why I say it's a low possibility that we're going to have a future life if an individual is cryopreserved is that there's a lot of scientific problems to it. There's very limited research in cryonics per se. I would say in cryopreservation and cryobiology itself, there's quite limited research at the moment. And in cryonics, there's virtually no academic research. There's some anecdotal evidence. There's some small studies, but very limited. So, for example, when we look at how much brain damage occurs during cryonics, we have very little data. I mean, Ramon actually presented some data, for example, and there is some data on structural integrity. We know fractures occur and so on. But we know little about molecular damage. I mean, I'm actually a cell biologist by training. We know very little about cellular survival, about molecular damage that occurs. There's something that I would suspect it's very high because I've frozen small amount of cells. I've cryopreserved small tissues. So based on that, extrapolating to cryonics, I would assume there's extensive damage, but I've never looked into it. And to my knowledge, there is no strong data on this. But so I think there's still a lot of question marks and a lot of problems regarding cryonics as a scientific approach. So what can we do at the moment? So I guess what I've been trying to do, on one hand, I've been trying to do some research on cryobiology. I mentioned a couple of examples of what we've been trying to do. And this is in spite of the fact that there's very little funding in the UK. I would say worldwide, there's little funding for cryobiology research. And there also seems to be little interest from students. So for example, my lab, we have quite a few graduate students, but they're mostly interested in age-related diseases like cancer, or they're interested in aging and longevity. It's relatively rare to have students that are interested in cryobiology and cryopreservation, although there are exceptions, and I'll mention one in a little bit. So in spite of these limitations of not having people, pairs of hands to do experiments and funds, we've been trying to push for some research into cryobiology. So I'll show you about two papers we've been involved in recently. So this one, I won't go into a lot of detail because we published this a few years back. So basically, this was a collaboration with Greg Fahy and others. But the goal of this work was to study cryoprotectant toxicity in vascular endothelial cells, which are important. So they're important cells in the vascular system. So they'll be crucial for cryopreservation of organs and for cryonics. And I mean, very succinctly, what we showed was that endothelial cells are resistant to ethylene glycol. They recover well from exposure to ethylene glycol. And then we also did some gene expression profiling using high-throughput approaches that I won't go into detail, trying to gather some insights into what could be causing the toxicity from cryoprotectant as well, ethylene glycol in particular. I mean, the paper we published already a few years back, so again, I won't go into details, but all the results are available online. And I mean, and this work was funded by Longicity. Now, more recently, we focused also on Drosophila. I'm not a biologist, Drosophila biologist, but we have several collaborators here. So for those who are not familiar, so Drosophila are these tiny fruit flies. They're widely used in genetics and biology studies. They have a limitation, which is you cannot cryopreserve them. So you can cryopreserve mouse embryos, for instance. You can cryopreserve C. elegans, roundworms, which we also use as a model. But it is not possible to cryopreserve fruit flies. Well, there's a paper written several years ago that says it's possible, but it's not been reproduced. I would stand by, again, there's some details I'm skipping. Happy to discuss in more detail, but I would stand by my statement that it is not possible to cryopreserve fruit flies, which is a big limitation actually for biologists to use this model. So what we tried to do was we tried to make advances in cryopreserving Drosophila larvae, which you see here on the right. And for this, we employed a permeabilization solution of, well, 10% aptame and 9% limonene. The timing of this is also quite important, and it's available in the paper that is now in press. But the point is that we created this permeabilization solution. And when you expose Drosophila to it, so this on the left here, you'll have larvae, Drosophila larvae. These are live and these are dead ones. And these are basically permeabilized with a dye, with a fluorescence dye. And as you can see, there's a very clear difference that when you permeabilize them before you put a dye in, the animals take up a lot more of the dye. So it is working in making these animals uptake the dye. I mean, you can do it from a more quantitative perspective as well. So you can look at controls, which I think is about 7 or 8% of the animals, 7 or 8% uptake of the dye, compared to I think something like 70% when you permeabilize them. So we show that this is working in permeabilizing this Drosophila, live Drosophila. And then what we wanted to do, of course, is we wanted to advance in the topic of cryopreservation. I mean, we did only one experiment for reasons that I'll come back in a minute, which was basically to expose the Drosophila larvae to a high concentration of cryoprotectants, namely ethylene glycol and DMSO. So what you see here is survival of the animals. So if you don't permeabilize them, all of them survive, because the ethylene glycol, the cryoprotectants don't go inside the larvae. If you permeabilize them without the cryoprotectants, most of them survive. But if you permeabilize them with a toxic dosage of cryoprotectants, then the vast majority of them die, which means that the cryoprotectants are going inside the animals. Now, these results are, so as I said, this is a paper now that is in press in the journal Fly, so it's available online and all results are available. This was work done by Andy Murray, who was a graduate student, master student in my lab, who was interested, so he was one of those exceptions, and one of those exceptions, a person who was interested in cryopreservation and was able to drive this work forward. As it was only a master project, so relatively short time, we were unable to continue it beyond this. So our goal next, if we have a student, so Alex is now a PhD student in Warwick, because I didn't have funds to for a project on cryopreservation. So he's not in my lab anymore. But if we have funds to continue this, the next step would be to try to cryopreserve the Drosophila larvae, as we can already show that they can uptake the cryoprotectants. So that's the work we've done so far. We're trying to push this. I mean, we just applied for a small project with Roman, who the previous speaker, on applying machine learning to develop cryoprotection methods for cryopreservation methods for the retina. So we're trying to push this. As I said, there are limitations in terms of funding and pairs of hands. So the other thing that we've been working on, and this is also with Roman and quite a few other collaborators, some of them are here in the conference, we wrote this quite big review of cryobiology and cryopreservation. The title is the same title of my talk, Winter is Coming, the Future of Cryopreservation. But the point of this review is, on one hand, to highlight the benefits of developing improve or enhancing the protocols for cryopreservation at the level of organ banking for organ transplants. That would be fantastic if we could cryopreserve whole organs, for the storage of engineered tissues and stem cells, and for, I'd say broadly speaking, for regenerative medicine. So we want to highlight that. We also talk about emergency medicine. I mean, there's some interest even from the Department of Defense of the United States in cryopreservation, and maybe even for space travel. So it's the paper aims to highlight this importance of developing research in cryobiology and cryopreservation, hoping to foster more research in this area. And we also discussed some of the limitations and challenges of current methods. It's quite a big paper. If anyone is interested, it's currently submitted for publication. If anyone's interested, feel free to drop me an email and I will send it to you. I mean, we're looking for feedback and opinions from others. I mean, ultimately make the argument that, you know, we need more research into cryopreservation. We need more research into cryobiology for lots of medical reasons. If this advances occur, then we will reach the point of having a method for human medical biostasis or cryonics. So that's the point of our argument. So we want to go from, so this would be traditional freezing, which develops the ice crystals, and this is vitrification. This was another figure that Alex Murray, who's also a co-author in the article, made. So that's something else we are working on. As I said, we're Roman and quite a few others. Jose Cordero is also in this conference, and a few others that are here today. The last thing I will mention is, this network I created a few years ago called the UK Cryopreservation and UK Cryonics and Cryopreservation Research Network. I mean, the goal is to advance research in cryobiology, including cryonics. I mean, focuses in UK, basically because I am located in UK. So that's pretty much the only reason, but it's actually composed of academics from all around the world. And really the goal is international. It's about fostering research in cryobiology and cryopreservation, including cryonics. So see what we can do to advance research in this area. I think one of the problems we have in cryonics is, I mentioned this if you see me give talks on cryonics in the past, I think one of the problems we have is the level of public perception or PR. I think we have a problem that there is a lot of skepticism from the public and for medical professions regarding cryonics. So really one of the keys to improve reputation of cryonics. So if there are news stories, then we can engage with the media on that. We can make statements. We can engage with other medical professionals. It's something I've done when they're critical of cryonics. I'll send them an email. I'll tweet them. I'll contact them and say, listen, why are you being so critical of cryonics? I mean, what are your scientific reasons? And a lot of times, I would say most of the times, there are no, because we don't have hard data on cryonics, there's no really strong scientific reasons for it. It's more, preconceived notions. So really trying to show that what we want ultimately is for medical biostasis is that to show that this is a high-tech, professional, and reliable approach. Now, I know we're not there yet, but that's where we want to go. And so that is the role of this UK Cryonics and Cryopreservation Research Network. And if anyone's interested in joining, again, feel free to contact me. So I guess what we want to go with, my goal in all of this is to create this bridge to the future that is medical biostasis. So develop the methods and the approaches that will allow us to have safe, efficient, reliable medical biostasis, which I think would be a revolutionary technology. And again, convince also the medical professionals and biomedical researchers that, this is a lofty goal. This is something we should aspire to because it would have tremendous medical benefits. So, so in summary, I've told you that, well, I think many of you are convinced that death is really, really, really bad. It's. I know there's people who will try to convince you otherwise, but really, death is the worst thing that can happen. Really, it is, and it is this eternal oblivion that I cannot even rationalize it. It would be wonderful if we could cure aging, but I don't think it will happen in our lifetimes. So if we cannot cure aging in our lifetimes, then we will die within our lifetimes. And so cryonics is this backup plan. Now, it still has a lot of technical limitations. There's a lot of problems. There's still a lot that we don't know because there's very limited research carried out in cryonics. But we can try to make progress. We can try to make progress in cryopreservation. I showed you our first protocol for permeabilization of live Drosophila larvae, which I think is a first step into cryopreserving Drosophila. I told you about our review of the cryobiology field. As I said, if anyone is interested, I'm happy to send it to you. And again, it's something else we're doing to gather attention and raise awareness and really show that this is a worthwhile field that we need to invest more into. Oops. And lastly, I told you about UK Cryonics Research Network to foster research in this area. So with this, I thank you very much for your time and attention. I mean, this is my lab. As I said, we mostly focus on aging, but I should highlight Andy here. This was a photo before the lockdown. This is, I think, from last year. So this is Andy here, who did Andy Murray, who did the experiments in Drosophila I told you about. And so thank you very much for your time and attention.
Joe, great. There are a few questions that I quickly would go through. So first of all, you kind of already discussed it a bit. Is there any update on the funding from US agencies on DARPA and others, both for cryopreservation in general, for soldiers and astronauts? How are they moving ahead with these funding?
So in UK, that's very limited. There is some funding in the US. So I know, I think people from the US, I think Greg Fahey is speaking later today. Right. I think they'll be better able to update you on that. In the UK, there's very limited funding for cryobiology, I would say. There's actually very few groups working on cryobiology. I mean, Roman is in the UK. So he's one of the very few people working on cryobiology in the UK. So I'd say it's very limited at the moment in the UK. I think what we're trying to do, so for example, in this project with Roman is, okay, so I do a lot of machine learning and do a lot of bioinformatics. So, machine learning is quite a hype topic at the moment. So, can we sell a project on applying machine learning to cryopreservation? Maybe that will, maybe that will be sufficiently interested to be funded. So we'll see. Fingers crossed on that. But that's what I've been trying to go. Instead of a project just on cryobiology, bring in some other more sexy element, like machine learning is very sexy at the moment.
Right. Awesome. Yeah. Then the question that kind of goes, I guess, towards your work that you do with the outreach program in the UK. Mm-hmm. Do you think that cryopreservation can be a mass market thing? Like what are hearing in the UK and from the people that you talk to?
That's a good question. A mass market. Well, I don't think it will be, incredibly popular like, machine learning. But certainly I do think there's a lot of room for it to grow. It's about making it mainstream. I guess that's the question. If you can have, people who have a disease, or we go in for, emergency medicine would be a good example, or battlefield medicine. If it becomes mainstream that when you have your life is in danger because of a gun wound or a disease, and you're just cryopreserved, then yes, that will become accepted and it will become adopted by the masses. But we're still quite far from that point, unfortunately. I do think that, there's a lot of very strange medical procedures that are done already. So I don't think cryopreservation in that sense is stranger or than some things that are already done. Open heart surgery it's kind of a pretty incredible procedure, if you ask me, for example, and it's done regularly. So it's a question of, one, having the biomedical advances that show this is reliable. And then at the level of public outreach and public communications, that showing that this is a worthwhile endeavor and that, it's backed by science and we can do it. And yes, then and at that point, it will be much more widely accepted.
Right. Thank you for that. Two more questions. First one being, do you think that research of Arctic ecosystems can provide valuable insights to advanced cryopreservation? I guess talking about animals that cryopreserve.
Yes, I think so. So yes, there has been some work on that. My lab hasn't worked on it specifically, but we've done, I've done some work on Arctic species actually, but not in a context of cryopreservation, more in context of longevity, because species on lower temperatures, they tend to be longer. So we've done some work on that. I would say yes, but the caveat or the limitation is that biology is complicated. So sometimes there are complicated pathways involved. Other times there's, I think there's been some studies showing that particular animals that can secrete some sugars into their bloodstream to protect against cryopreservation. So I would say yes, it's absolutely it's a topic that would be very worthwhile to explore further, because we can learn a lot from biology. There's lots of examples of innovations in medicine that we learn from animals. We learn from evolution. We learn from the natural world. And I mean, just if you're interested in more details, I actually published a review on this in EMBO Report in 2015, which briefly mentions this cryopreservation as well. So yes, absolutely.
Right, and then last questions for now. Could you elaborate on the arguments why fruit flies can or cannot be cryopreserved?
So that's a good question. I've never tried to cryopreserve fruit flies myself. So I can tell you the larvae, for instance, they have a particularly waxy cuticle around them. So that's the reason why they're not easy to cryopreserve. So what our method does is permeabilize that waxy cuticle to different agents, to cryoprotectants, but also, I mean, this method can be used for toxicology, for drug testing, can be used for a lot of other applications other than cryobiology. So that's the problem of the larvae. I'm not entirely sure about adult fruit flies, why they cannot be cryopreserved. I guess it will be, again, difficult to permeabilize them to the cryoprotectants. I would think that's the limitation. So there are a couple of papers. I think there's a Nature paper from like the 70s talking about cryopreservation fruit flies. But what everybody I asked told me is it doesn't actually work. It's not reproducible. So we just don't have a method. There's not been that many research on cryopreservation of fruit flies, but what everybody works on fruit flies they've asked told me is it would be brilliant if we have a method. Because for example, when we create a genetically modified strain, let's say a long-lived strain for studies of aging, we just have to keep the animals alive all the time. So we have to keep them breeding. We can't cryopreserve them, put them in the freezer, say there's a pandemic. We do work in C. elegans and cells. We just freeze our stuff and that's it. Right. But I assume my colleagues who work on fruit flies, they have to keep going to the lab. They have to keep feeding those worms because they have to keep reproducing. You can't just freeze them. So it is an important bottleneck of research. And I think our approach, I think it's a first step. I mean, that's the thing. A lot of these are first steps. It's incremental into developing cryopreservation for Drosophila.
Perfect. Thank you so much. Unfortunately, we don't have more time for questions right now, but we would then later on tonight or in the evening have actually a few more. And sorry for every attendee who didn't have their question answered, but there's another chance at the end of the talk. João, thanks a lot.
Thank you.